And kicking us off this morning, we have Carsen from Janelia, who will be talking about superhuman generalization for cellular segmentation. Thanks, Carsen.
Can you hear me okay? Cool. Hi, everyone. I'm going to be talking about our latest version of Cellpose-SAM. I'm a group leader at Janelia, and Marius Pakisar is also a group leader here, and this is a collaboration with him. So what is cellular segmentation? You might have an image like this, and you want to draw outlines around all of your cells, which ultimately become these pictures of cell masks, where basically you label the background with zeros, and then 1 for all the pixels in one cell, 2 for the next cell, and so on. And so I think most people here know why you would want to segment cells, but basically, like, you want to track cells over time, you want to quantify RNA expression, you want to quantify neural activity. There's lots of applications for segmentation. And so we created Cellpose as an algorithm to perform this task. And so how Cellpose works is you take a cell, and let's say you have this annotation, this outline of the cell. We create these flow fields which create this dynamical system for each cell, where if you were to run this dynamical system with all the pixels would converge to the center point. So you have Y gradients and X gradients here, which we represent with a circular HSV color map like this. And so this works for a wide variety of cell shapes. These flows can turn around corners and work on these non-convex cellular shapes. So we trained Cellpose, we trained a neural network to take as input cellular images and output these flows, and also a cell probability map of zeros and ones. This is the original Cellpose. We used a UNet and then added residual blocks for more capacity, and also these style vectors, which we use for size estimation. After you get the flows, the post-processing, so the flows come from the neural network, and then we have post-processing steps. So this gradient tracking is basically running that dynamical system I described, where you run every pixel with these flows, and all the pixels that converge to the same point, they get assigned the same cell ID. So you can see here now, if we run these the flows, we get an image of masks that looks like this. So Cellpose works across a wide variety of cellular images. And so this is shown here from our recent Cellpose 3 paper. The ground truth is in purple, and the model is in yellow. And so it can work on these really long shapes, too, and a variety of shapes. So we wanted to know, what we were thinking was what's next? Like, is this the best that we can possibly do with segmentation? So there was this recent cell segmentation challenge at NeurIPS 2022. And they compared, they had some winners of the challenge, and they compared to Cellpose. And so the F1 score is like your accuracy, higher is better. And so Cellpose is over here, and the best performing models over here. And so we looked at this, and we thought, okay, well, maybe there was some big improvement that happened in cellular segmentation. When we dug a bit deeper, we found, so this winning model called Meteor, they used a segformer backbone, and then they used the Cellpose loss tracking and extraction code, the same as what we have. And so we thought, okay, well, maybe this backbone is important. So we dug a little bit deeper into the paper and the challenge. And so here's an example of validation image from the challenge. And this looks like something that Cellpose might be able to segment. This is the model that they trained in their paper to segment this image. And again, ground truth is in purple. You can see Cellpose missing a bunch of these cells, which suggests maybe they trained Cellpose incorrectly. And indeed, if we train Cellpose on the challenge data, we do fine on their validation images. And we can summarize this with this F1 score across all the validation images. Here's for Cellpose. Here's for Meteor. Here's the Cellpose they provided in their paper. And in fact, if we take our Cellpose model and replace that backbone with the segformer, we don't perform any better than any of the other models. So you can look at the paper if you're interested in why there were these differences. But basically, this suggests, well, maybe we're kind of hitting this upper bound of performance with these models in terms of segmentation accuracy. And so is it actually possible to do better? So one way we benchmark how well we're doing on segmentation is to look at like inter-annotator consistency. So let's say you have annotator 1, which is shown in gray here, and then we look at what annotator 2 drew on the same image. And so there will be some false positives. So these red dots are cells that annotator 2 drew that didn't exist in annotator 1's annotations, or there'll be these X's, which are these false negatives, which are cells that annotator 1 annotated, but annotator 2 didn't annotate. And so we can compute the error rate between annotator 2 and 1. So the number of errors, the false positives plus false negatives over the true positives plus false negatives. And we can get some kind of a sort of what some might call a lower bound for the error rate we can get for segmentation. If we compare that to our Cellpose models, the Cyto3 and the segformer model, we see we're pretty close to this supposed lower bound. But we don't actually think this is the true lower bound because if you think about it, each annotator 1 and 2 will randomly make some mistakes, let's say 10% of the time. And then you're comparing these two noisy estimates to each other. So you can imagine if we had a human consensus, let's say we had 100 humans label this image, and that's this human consensus shown in black, we'd actually get fewer errors if we compare annotator 1 to human consensus than when we compare annotator 1 to annotator 2. We'll get fewer false positives because the human consensus will have fewer errors, for example, than annotator 1, and we'll also get fewer false negatives because we'll have fewer of those misses in the human consensus. We'll have fewer false positives in the human consensus, which are counted as false negatives for the annotator to annotator score. So this error rate is approximately half if you think of an annotator 1 to human consensus. And so we can show this error rate here. This is kind of our estimate here, that we should have about half the errors if we look at this human consensus performance. So this is really our true lower bound, we think, for these algorithms. And we're not reaching that mark with these algorithms here. So maybe indeed we could do better. And so looking into machine learning, there's this state-of-the-art algorithm for segmentation, Segment Anything, which has been trained on over 1 billion masks from 11 million images. And so they train this model to do this segmentation task that you can see here. And they train a large vision transformer model to do this task. They have multiple sizes. And so we looked at, for example, there have been recent papers that have adapted SAM for microscopy. So there's the micro-SAM paper and the cell-SAM paper. And so we looked at how well do these models perform on the Cellpose test set. And we found that they did not outperform Cellpose, and they didn't go lower than this inter-annotator lower bound that we described here. So we thought, okay, maybe we should try to adapt SAM and basically combine SAM and Cellpose together to make a better model. So we take Cellpose, where we're taking an image and predicting the flows. We take SAM, which has this image encoder and this prompt decoder, and we basically take the image encoder from SAM and use that instead to predict the flows. And so we also modify the image encoder a bit, so we run on 256 by 256 images rather than 1024 by 1024 images. We also run on 8 by 8 patches instead of 16 by 16 patches. So, and then after this image encoder predicts the flows, we have our post-processing steps as well, the gradient tracking and the mask extraction, like before. Okay, so how do we retrain SAM for Cellpose? So we take the SAM weights that were trained on their task with their prompt decoder, and we retrain it as a foundation model by converting their masks from their training set into these flow representations that we use in Cellpose. And so we used over basically 2.6 million of their training images to retrain Cellpose-SAM as a foundation model to do this task. And then we fine-tuned this model on cell data sets. So we have around over 11,000 images that we've combined across many different modalities from many shared data sets. And that's kind of our base Cellpose-SAM. And then optionally, and I'll show results on this later, you can fine-tune this model on user data with 1 to 100 images if you want to specify it make it specialized for your task. Okay, so how well does Cellpose-SAM perform? it performs better than these previous models. So here's our error rate for Cellpose-SAM. And so we're below this inter-annotator error rate, and we're much closer to this, what we think is this human consensus error rate here. We can also quantify this as accuracy, basically just kind of, which is inversely related to that error rate. So average precision, higher is better. And you see Cellpose-SAM in yellow here outperforming these other models. And I'm showing some example images here. The ground truth is in purple again, and the output of the algorithms is in the respective colors of the algorithms. And so you can see Cellpose-SAM on these example images outperforming another foundation model for cellular images, CellSAM. Okay, so you might be worried that Cellpose-SAM is going to be really slow. So this is a number of parameters in our Cellpose Cyto3 UNet, and this is a number of parameters in Cellpose-SAM. But fortunately, modern GPUs have these tensor cores, which make matrix multiplication super fast. So actually, our Cellpose-SAM model is faster than our previous models, also because it doesn't have to have this size model in addition. And it's actually faster than the other SAM models that have been released in the field because we use these smaller image sizes of 256 by 256 rather than CellSAM uses 1024 and MicroSAM uses 512. And so I think for most use cases, you'll see like a 512 by 512 image is running in 0.2 to 0.3 seconds, which for most cases is sufficiently fast. Okay, so we also trained Cellpose-SAM to make some quality of life improvements for users. So we trained Cellpose-SAM to have channel order invariance. We basically permuted the channels during training randomly. And basically the performance of the model is equivalent across all of these permutations of channels. We also trained the model to have size invariance. So an example image, like you can see here, the cell diameter is at 30 pixels, and then we could upsample it to 90 pixels or downsample so the cell diameters are on 8 pixels. And so we trained the model on all of these different sizes and successfully achieved size invariance. So this is the average precision score across all of these different sizes. And you can see it's substantially higher than the previous models. One nice thing about having this size invariance is you can put a large, an image that's quite zoomed in into Cellpose, and if you have these kind of finer structures, you can actually see that the outlines from Cellpose-SAM are more accurate looking than the human annotator, because the human annotator was actually annotating at this 30 pixel resolution. So this could be really helpful for like long dendrites or other fine structures that you need to segment. We also trained Cellpose-SAM to have noise robustness. So we added Poisson noise during training, and we found across these noise levels, low, medium, and high, Cellpose-SAM is able to directly segment these noisy images much better than previous models, and actually just as well or better than our our restoration model from our recent Cellpose-3 paper. So basically, we train models to do things like denoising images. So you denoise the image, you get the denoised image, and then you run the segmentation on that in this two-step procedure. And actually now with Cellpose-SAM, we can basically, you can just run the model once on your noisy image and get out the segmentations, which is nice. Additionally, we trained it for robustness on other degradation cases, so like blurring. You can see Cellpose-SAM performing as well as our restoration models with the previous model applied. And this also works for anisotropic images. So this is useful for, we're already applying it in 3D data. That seems to be quite useful, and pixel size robustness too, like downsampling and upsampling. We also found that we can use Cellpose-SAM for fast fine-tuning. So here's EM data. So Cellpose-SAM has never seen any EM data in the training set. And so the output of the algorithm on these example images from these two data sets, you can see is just kind of a bunch of random ROIs. It's not really matching these purple ROIs. It's supposed to predict just the mitochondria of the EM. We can quantify that as this average precision as a function of the number of training ROIs. So we're at 0 here because we didn't train on any of this data. And then we can take some annotations from the rat EM and from the human EM. We train a model on each of these. So one model for I'm training the models, one model for mito-EM rat and one model for mito-EM human. And you can see that these fine-tuned models, one fine-tuned on 7 ROIs and one fine-tuned on 4 ROIs, already does substantially better. So you can see this average precision goes up. And I'm summarizing it here as an average over the test images that we have. And so you can see this big jump in performance just with one. Basically, this is one example image that the model's been trained on. And then we add more images, and you can see this performance goes up quite quickly. This is a log scale, so we get a lot of performance improvements with just basically 100 training ROIs with Cellpose-SAM. And we can compare to our previous model. So we take Cellpose-SAM 3, basically out of the box. It also doesn't do very well. But as we fine-tune it, you can see it. In this case, it eventually reaches the same performance as Cellpose-SAM, but in this case, it actually never reaches the same performance as Cellpose-SAM. And it's also going up substantially slower. So it's going to be slower for you to do human in the loop here than if you're using Cellpose-SAM. And the training of Cellpose-SAM is similar to the training time with the Cellpose-SAM 3, or faster, because we use fewer epochs for training, fewer iterations. So this is all doable, basically, human in the loop, like if any of you have used Cellpose-2 before for human in the loop. We also compared on a data set where both of them kind of have some performance out of the box. So these are fluorescence images, and Cellpose has seen fluorescence images. But you can see out of the box, Cellpose-SAM does better on these lateral root images from Walney et al. And actually, this improvement in performance is maintained as we train on more and more ROIs. We also trained Cellpose-SAM as a foundation model for other tasks. So there's this example, Panoptic segmentation challenge, MONUSAC, where they had a bunch of H&E images with different cell classes. So these 4 different cell classes, and this is the ground truth. These are the ground truth annotations for the test set that they have, and the colors correspond to these cell classes. And so we retrained Cellpose-SAM with an additional output for cell classification. And we were able to, basically, this is what the segmentation looks like with the classification. So the segmentations are dashed, and the color of the dashing is basically the cell class that Cellpose predicted for each of the cells that it segmented. And so you can see good alignment between the true classes and the predictions from Cellpose-SAM. And if we can look at the average precision score across all of these classes, and we're doing as well as or better than these previous winners of this challenge. So just to summarize, we found that Cellpose-SAM is more accurate than this interhuman or interannotator agreement. It's almost as accurate, we think, as the best possible estimate for how well you can do. We made some quality of life improvements, the size invariance, channel order invariance, this noise, blur, and anisotropy pixel size invariance. So we're hoping, like, as users, this should make Cellpose much easier to use. You don't have to think about what kind of degradation did I have in my image or these other things. Where should I put my nucleus channel or other things like that. Also, if you have 3-channel images that you want to retrain Cellpose on, it's kind of already seen cells in all of these channels, so it should be easier to retrain your model with 3-channel images if you want to use more information, maybe for your training, for example. We also improved the fine-tuning on segmentation and other tasks, so you can fine-tune this model much faster. It's fast enough to train human in the loop, so you can, ideally a biologist can do this in an afternoon. And it's fast for a foundation model. And yeah, that's it. Thanks everyone for listening. Thanks to the organizers and all the 4DCP group leaders and Janelia. I'm lucky to be able to collaborate with a lot of you, and thanks for your time. Oh, and I have stickers if anyone wants some.
Carsen, this is great. Just a technical question. So when you train the vision transformer models, you said you use 8x8 patches instead of like 16 by 16 for better global attention. I think that's
Oh, sorry, I didn't mention I forgot to mention the global attention thing. Yeah, so the global attention is just saying if the patches interact with each other or not in each layer of the transformer. And so in the default SAM, actually they only have global attention in the VITL for 4 of the layers, and we actually do it in all the layers, and we find that helps more. So you're constantly using all of the context from the image.
I see.
That will happen regardless of patch size, though.
Okay, but for the patch size, do you compare different levels? Like, do they perform differently?
We've tried 4x4 and 16x16, and this we find works the best. That we've tried. Yeah. But it's a good question.
All right, we have a question on Zoom from Samsara Upadhyaya. Really interesting. I'm wondering how the detection of debris works for Cellpose-SAM. Thanks.
The detection of debris?
Yes.
I think it will depend on what the debris looks like. Ideally, I'm hoping, I'm optimistic about it because it was really fast here to learn to ignore everything else, like to go for just the mitochondria in these images. So I'm hopeful with a couple of images of debris, it shouldn't be too bad.
Hi, very nice work. I'm wondering, so a lot of the images are, I think, in 2D. So how well does it perform in 3D? Is the flow field also in 3D? And the second one is more specific to neurons, I guess. If you have a neuron that has long processes extending out, how well does Cellpose sort of segmenting those processes like axons and dendrites?
Yeah, that's a really good question. So I think it will work better than the previous versions in segmenting longer things. But if you end up starting to get overlaps between objects, then things are going to be tricky if you only have 2D. In 3D, those things maybe can be resolved. And then for the 3D case, the way that Cellpose works is we take X, Y, YZ, and XZ slices, and we run Cellpose in 2D on these different, on the XY views and the orthogonal views of the image. And then we get flows in each of these cases. So like in XY, we'll get XY flows. In XZ, we'll get Z and X flows. And then we average them, and we get flows in 3D, and then we run the flows in 3D. And we find that works quite well in most cases when things are not super complicated in terms of shape.
So a chaser question. So
yeah.
If, let's say you're imaging, like image acquisition, like most of the optical microscopy setup, I guess the exo resolution is way worse than XY. So in that case, basically, do I have to like downsampling the XY or?
No. Actually, so this is a supplemental figure in the Cellpose 3 paper, and actually, I've started doing it for this model too. Is you can run it on the blurry images. You can either do restoration to basically you upsample in Z instead and restore, or this model seems, what I found, it seems to be working out of the box well on anisotropic images. Yeah, it's a good question.
Thank you.
Maybe I can ask a question. So, just following up on that, I guess maybe you can comment a bit, Carsen, on what it would take to make like a true 3D version of this. Like, what would be involved in that?
Yeah, basically the reason we think Cellpose works so well is because of the diverse training set, and so it's really getting a diverse training set in 3D, and so we're starting to work on that now, and with you and other people, and basically if we can get a really large data set in 3D, we can train this a generalist model ideally also in 3D, in full 3D. I think in a lot of cases, I think we can bootstrap quite a bit if the segmentations in 2D are good to do some human in the loop in 3D. And I think also there's a lot of cases where the 2D to 3D is good enough, particularly if you take into account these anisotropy issues.
Hi, I wanted to know so if you had a unit instead of a vision encoder, but it had 307 million parameters, would you think that you can get the same level of performance? And so my question comes from how important is model architecture versus diversity of the training data set?
That's a great question. We've also tried larger UNets. They're much slower, so they've never been that big. So I think transformers have this real advantage that they're much faster and can have a really large capacity compared to the UNets. So it would take a really long time to train a UNet with 307 million parameters. So we haven't tried that. So I think that is really, it is just an issue of how well can these models scale to larger sizes or not. And that's the advantage with the transformers.
Thank you.
Yeah, it's a good question.
Given that we're in like the 4D talk, and also because SAM also allows for tracking objects, are you thinking of extending Cellpose towards tracking also?
So it's definitely an interesting idea, but we haven't tried it yet. The SAM2 tracking data set, I think, is a bit smaller, but maybe it's...
No, SAM, I think it's hard to take SAM out of the box, indeed, because it's like limited in the number of objects it can track. So you need to do something smarter to kind of like think how to extrapolate, but just because...
Yeah, no, I think it is a good point. I think these transformer architectures could be very good for cell tracking too, but we haven't tried it.
Okay, if there are no more questions, let's thank Carsen. Thank you, Carsen.